All right, so just as a reminder to what we were talking about, we began to look at glycolysis, and we were working through the different reactions of glycolysis. So we're going to finish that today. And then after that, we're going to start moving into what do we do after glycolysis. So let's continue on that. Luckily, in terms of mechanisms, we're not going to delve deep into the mechanisms that much. If you look in your textbook, there's a ton of mechanisms in your textbook for every single reaction. The only ones we go through these live sessions are the only ones I'm going to really ask you to know. So we don't need to know, 10 mechanisms. We need to know just a couple. All right. So we ended with this question: why do we need to convert our 6-member ring to 5-member ring? And it's just the ease cutting into three carbon compounds. All right, so let's move on to the third reaction. Phosphofructokinase, PFK. And I'll just say this now. So if I ask you a question on a test and I ask you for an enzyme name or a substrate name, you can say the full name. I will also accept PFK. So if you want to remember the names as like PFK, F6P, FBP, that's totally fine with me. All right. So in the last reaction, we made our fructose 6-phosphate. Our next reaction uses a kinase. Again, the easiest way to really learn this stuff and to help you in the future is to really look at these enzyme names and break down what they mean, right? So phosphofructo, well, fructo is what we're working with, fructose. Phosphofructo means I have a phosphate on my fructose. Phosphofructokinase, a kinase is an enzyme that adds a phosphate. So if I was going to say PFK's name in like an English sentence, it's an enzyme that adds a phosphate to a phosphofructo group, phosphofructokinase. So it's adding an ATP to make fructose 1,6-bisphosphate. Here, the name of bisphosphate is important. Throughout your career so far, I'm sure you've heard diphosphate as in ADP, adenine diphosphate. A diphosphate is when two phosphates are connected. I guess that's an oxygen in there. A bisphosphate is when you have two phosphates on a single molecule, but they aren't connected. So that's the difference between a bisphosphate and a diphosphate. The reaction here, very similar to hexokinase we saw on Tuesday, where we're adding ATP to glucose, or a phosphate to glucose rather. Here we're doing it to F6P. We use magnesium, and it's the same like mechanism as we saw for reaction 1. Magnesium is shielding the negative charges of our ATP, allowing for that attack to put a phosphate on. We're going to look today about PFK and its role in maintaining glycolysis speed, and it is the most important step of glycolysis. It is the RDS, or rate determining step. So reaction 3, we're going to talk a lot about reaction 3 in detail because it is critical for turning on and turning off glycolysis. Because it's so important, we can regulate that enzyme allosterically. If you remember the word allosterically, you might not from Biochem 1, it's basically meaning that other molecules will interact with the enzyme and it can either turn it off or turn it on. That's more or less what allosteric means. I interact with the enzyme to modulate its ability. So this enzyme can be turned on by AMP, which should make a lot of sense, right? when ATP breaks down, it goes to ADP, and then it can go to AMP. If you have a high level of AMP in the cell, that means you have low levels of ATP. High levels of AMP means you are low in energy. Therefore, to make more energy, turn on glycolysis. To turn on glycolysis, activate the enzyme that controls glycolysis. So that's why AMP can affect PFK. While it'll be inhibited by ATP, this is the opposite logic of AMP. ATP, if you have a lot of it inside the cell, that means you have a lot of energy. Therefore, don't waste glucose by breaking it down. You already have a lot of energy. So ATP will turn off PFK. Citrate, same idea. Citrate is made in the citric acid cycle. If you have a lot of citrate, then it's going to say you have a lot of energy already made or going to be made very soon. So stop wasting glucose through glycolysis. This is something we talked about in the first two lectures, feedback inhibition, where citrate is made from the citric acid cycle, which is made from glycolysis. So it's actually going back up the chain to inhibit PFK. So any questions about the ideas presented here? Like I said, we'll go into PFK quite a bit this semester. All right, moving on then. So reaction 4, we take our fructose bisphosphate, and now we are going to cut it in half into two different molecules, DHAP and GAP, dihydroxyacetone phosphate or glyceraldehyde 3-phosphate. And the enzyme that's doing this is Aldolase. And the numbering gets a little wonky here. If we look at our numbering of fructose 1,6-bisphosphate, 1-phosphate is number 1, then the carbonyl carbon is 2, all the way to 6. The numbering goes, what was 1 of our fructose bisphosphate is now carbon 1 of DHAP. And you can actually go further if you want to go. So that should be glucose. If you go back and look at the numbering of glucose, atoms 1, 2, 3 of glucose are 3, 2, 1 DHAP. Atom 4, 5, 6 of glucose are atoms 1, 2, 3 of GAP. So we can, by doing experimentations, scientists have discovered this is how the enzyme works. And we take a second and think about that. It's really amazing that people can get that level of detail about one sugar molecule to know when it's split in half, where do each carbons go? So if we look at this mechanism, it is an aldol cleavage. So we're going to look on the mechanism here on the bottom left. So this is what we're looking at here on the bottom left. That's just a general molecule, but it is in the shape more or less of FBP. It's like this would be more or less like carbon 2, carbon 3, carbon 4. It's not quite the same molecule, but you get the idea. So the first thing that happens is base catalysis, where we take the hydrogen from the carbon 4 hydroxyl group. That will create a negative oxygen, which will form a double bond with the carbon. And the carbon will split. Carbon 4 will break from carbon 3. So this is product 1 down here. From there, we have a resonance structure enolate intermediate. Remember, an intermediate is a molecule that is stable in a reaction. So we can actually measure the amount of enolate if we were to do this experiment. So this is a stable intermediate. And what happens is that we need protonation. So more or less an acid. So at first we have water acting like a base. And then our third step, we kind of have water acting like an acid because we have this carbon group that is negative, has some negative characteristic to it because of that resonance structure. Carbon is really unstable if it's charged. So it will get a proton from water and get our second product. And so if we were to look at our two reactions up top, DHAP is more or less product 2 from our mechanism. GAP is product 1 from our mechanism. So that's how we can look at the picture on the bottom and connect that with what's going on the side. So here's a question, and it kind of goes back to some of the fundamentals we talked about in week 1. So this reaction, Aldolase, it's incredibly unfavorable. It is not spontaneous at all because you are breaking a carbon-carbon bond. That's not favorable. It has a delta G of plus 23.8. That's a massive number. That's very, very hard to get over. But this happens in every one of your cells. And it's happening right now if you had lunch too, probably. So how does this reaction happen? What mechanism do cells use to pull this reaction forward? That is, how do cells make sure this happens? Coupling, yeah. So the idea that we're going to see here is that when we make these 3 carbon products, we need to take them away very fast. If we have a buildup of these products, this reaction won't go forward. But to compensate for that, we have another reaction that's going to take these 3 carbon compounds away so we can continue this very unfavorable reaction. And we'll see that. We're going to look at a table today, or maybe next week, Tuesday. Keep in mind when we look at not prime, or I guess I should ask, what does not prime mean again? Does anyone remember what not prime is? Standard conditions, that's correct. Not prime means this is the energy under standard conditions. And if you remember, standard conditions means everything's at 1 molar. So our products and our reactants are all at 1 molar, and we solve for delta G not prime. Nothing in the body's under standard conditions. Those are just like fake numbers scientists use to get relative favorability, unfavorability. So we'll see that in the actual, in our cells, these concentrations are actually super low, which actually makes delta G not as positive. What is the difference of delta G not prime and delta G not? So this should be biochemistry standard state. I believe this is chemistry standard state. The big difference that I know of is, I want to say like pH might be one. Also, I think the concentration of water is one off the top of my head. Because for biochemistry, we say pH is 7. While in delta G, we're saying the concentration of H is 1. So big difference there. I think that's a major difference now that I think about it a little more. So here we're saying concentration of hydrogen is 1 times 10 to the minus 7 versus chemistry standard conditions, it's 1, which kind of makes sense because when you're doing chemistry, there's no guarantee you're working with water, while biochemistry, it's almost like a 99% chance you're working with water, and it's going to be at a neutral pH. So when we do our calculations, we're going to make sure we're at a neutral pH. There might be another difference I'm not thinking of. I would have to relook at those. I'm so used to biochemistry standard state at this point. All right, reaction 5. So we have DHAP and we have GAP, but our next reaction is simply converting our DHAP to GAP. This is done by TIM, Triose, Phosphate, Isomerase. Again, we can look at the words and figure out what this means. Triose, a 3-sugar, triose. Phosphate has a phosphate group attached to it. Isomerase. So an isomerase is an enzyme that simply moves around functional groups. That's what it does. So again, it's in the name. 3-sugar phosphate moving groups. TIM, triose phosphate isomerase. We have another intermediate here going from GAP to DHAP. So here's, or rather DHAP to GAP. We have this enolase intermediate. But moving forward, we only want GAP. So simple reaction right there. And this more or less ends phase 1 of glycolysis. We took 1 glucose, we incorporated 2 ATP, but we have gotten nothing out of it except 2 GAP molecules. So that is the end of phase 1 of glycolysis. So the next question I have, why bother doing this? Why don't you just put GAP and DHAP through glycolysis? Why do this weird conversion? Why, evolutionary speaking, would we want to do this conversion? Good critical thinking question right there. You don't want to have any hypotheses. Well, let me help out your thinking a little bit. Remember, enzymes usually only work on one substance. They're very, very specific. Is it easier to deal with one type versus 2? Yes. So I'm looking as to why that is. And it really has to do with enzymes, right? So enzymes can only usually work on one substance. There are some enzymes that work on multiple things. Most of them don't, though. So let's do the thought experiment of if we never did this step. GAP DHAP. The end goal of glycolysis is pyruvate. So if we had 2 different 3-carbon compounds, we would need an enzyme 6 for this reaction, enzyme 7, enzyme 8, enzyme 9, enzyme 10. That's more or less what it takes to go from GAP to pyruvate. Well, we would also need probably enzymes like that. So I'm just going to name them A to show that they're different. Who knows how many steps this takes? Let's just say it takes the same number of steps. So you would need a whole different set of enzymes to work on DHAP. So one, it just simplifies things. If I make everything GAP here, now I only need these enzymes to go to pyruvate. Making two enzymes that do the same thing, more or less, inside the cells, kind of a waste of energy. Also regulation, right? If I just have one pathway, I can just regulate that pathway. If I have two pathways, I have to regulate two pathways. It makes it much harder. So the idea is it is simpler to just do the conversion right here. You have one set of enzymes. It is much more efficient, yes. Life, for the most part, goes through the path of least resistance. I'm sure if you've heard that phrase before. It's the same thing with biochemistry, right? Enzymes aren't going to be complicated when you look at their mechanisms, and if you understand chemistry, it's a lot of logic, because they're programmed, they're made to do easy things. So if you had two organisms that are competing, right, and they have the same energy source, they have the same amount of food. Well, if an organism doesn't do this conversion, they more or less use more energy to make enzymes to get to the same endpoint. So they're wasting energy doing this. Therefore, over the long run, they're less fit. And so they're more likely to be outcompeted, if you want to go the long view of evolution with that. So it's just energy efficient. It's easier. All right, let's go to reaction 6, where we're making a high energy compound. So far we've only put energy in. Now in the next steps, we're going to get energy out. So we're going to take this GAP and we're going to phosphorylate it and form 1,3-bisphosphate glycerate. The enzyme that is responsible for this is a dehydrogenase. All right, so another little keyword for you. When you see dehydrogenase, that means we're going to do a redox reaction for the most part. Dehydrogenases more or less do redox reactions. And that's in your other clue for a redox reaction. When you see NAD and NADH, 100% a redox reaction. That's what NAD does. Remember, we talked about NAD. It is an electron carrier. All right. Now, we are making a very high energy compound. In fact, this is higher energy than ATP, right? So we're making a compound that has more energy in it than ATP. How does the body do this, or how do our cells do this? Well, one of the reasons this happens is because oxidation of this carbon, this aldehyde, is very favorable. That carbon really wants to be oxidized. And so some of the energy that we're putting into making this 1,3-bisphosphate glycerate, we're getting back for free because we're doing something that chemically wants to happen anyways. We're oxidizing this aldehyde that wants to be oxidized. So we're kind of piggybacking on that to make a very high energy compound. If you notice here, we're taking in inorganic phosphate. Anytime you see PI, another word for that is inorganic phosphate. It is just simply a phosphate molecule that is floating around in water, right? Imagine ATP, you break off one of those phosphates, that's PI. So we're taking an inorganic phosphate that's just floating around anyways, sticking it onto GAP to make 1,3-bisphosphate glycerate. Here, what is being oxidized and what's being reduced. I will just do this relatively quickly. So hopefully you remember how to do our redox numbers. Oxygen's always -2, hydrogen's always +1. This carbon therefore must be +1. Here for the phosphate, here's a little way to speed this up. It's -2 overall. So everything added together is just -2. You don't have to do individual oxygens and phosphates. You can just say this group -2. This oxygen -2 +4. So carbon went from +1 to +4. Therefore, is that oxidation or reduction? Does this carbon get oxidized or does it get reduced? If it goes from a +1 to a +4. Ox. Always remember, oil rig. Oxidation is loss of electrons. Reduction is gain of electrons. An electron is a -1 charge. So if I lose electrons, I become positively charged, right? So this is oxidized. Therefore, this was reduced. NAD, and you remember we saw this. It gets that weird hydrogen with 2 electrons on it. That's why it becomes NADH, and that's why it loses its charge as well. So there's a redox for you. And like I said, when you see dehydrogenase, think redox reaction. All right, reaction 7. We're going to make ATP. Finally, after 7 reactions, after spending 2 ATPs, we get an ATP out. Granted, in the last reaction, we made NADH. That's going to be important when it comes to oxidative phosphorylation. So that NADH is like stored energy that we can't spend yet. Reaction 7, we're immediately getting energy out. And just as a reminder, we are looking at 1 3-carbon compound here, but for every 1 glucose, we are getting 2 of these. So we are getting 2 ATPs out. So by reaction 7, we've already gotten back all of the money we put into glycolysis. We've already gotten back all the energy we spent to get to this point by reaction 7. Right now, we are in the black because we have that NADH waiting. If we can't use that NADH, at least we're neutral right now. All right, so this reaction is we're taking this phosphate from 1,3-BPG, adding it onto ADP to make ATP. This is carried out by a kinase because that's what kinases do. They move around phosphates. Phosphoglycerate kinase because we're working on a phosphoglycerate. See, the thing about biochemistry that's really great is that a lot of the names make sense. Once you understand the language, once you look at the words and get past their long nature and start to break them down piece by piece, it does make a lot of sense. So this reaction is coupled to the previous reaction. So remember, I said we're making this very high energy compound. So that has a positive delta G. Because even though it is favorable to oxidize this aldehyde from GAP, overall it's still positive delta G, so not spontaneous. But we use it directly in the next step to make ATP, very negative. Coupling. This is another example of coupling. We take the product from reaction 6, go right into reaction 7. Overall, when you combine reaction 6 and reaction 7, that is the energy you get out, minus 12.1 kilojoules per mole. This is a spontaneous reaction for making ATP and NADH. All right, so up to this point, take a second break here. Any questions anyone might have about anything? All right, then we'll move on. So how does 7 pull 5 forward? We kind of just answered that. We said 5 is really unfavorable, and I already kind of have a question. It's coupling, right? 7 is coupled to 6, 6 is coupled to 5, 5 is coupled to 4, so on and so forth. It's like a giant treadmill that we keep going and going and going on. Everything is connected. So you can have some unfavorable reactions there, as long as when you combine all 10 reactions, they have an overall negative delta G. So coupling is the answer to that question. Reaction 8, and it's a little cut off, so I'll redo the words. So 3-phosphoglycerate, 3PG. This is 2-phosphoglycerate, 2PG. So reaction 8 is called a mutase. A mutase is similar to an isomerase. A mutase is simply moving a functional group around. So phosphoglycerate mutase works on phosphoglycerate, and it's just moving a group around. All we're doing is we're moving phosphate from carbon 3 to carbon 2. Tells you in the name that that's happening. You go from 3-phospho to 2-phospho. Net neutral. Delta G is roughly 0. So either of these compounds, you either put the phosphate on 3, put the phosphate on 2, doesn't matter all that much. But we need to do this for reaction 9. So this is reaction 8's a giant prep step. Don't really get anything out of it other than a different compound. Reaction 9, we are making our second high energy phosphate compound. So we had 2PG here, 2-phosphoglycerate, and we're making phospho enol pyruvate, or PEP. So here we are taking away a water. So that's why I say dehydrated. Water comes off. And we are making a double bond between a carbon and a carbon group, forming an enol. So those of you who really studied hard in organic chemistry, really committed yourself to learning those functional groups names, congratulations. All that hard work is going to pay off when you look at something like enolase and phosphoenolpyruvate, and you can tell that there's going to be a double bond in there. Enol. En is double bond. All more or less means like alcohol, more or less. Those of you who didn't, I mean, you can still learn it just fine. So enolase will make this compound. Unfortunately, enolase doesn't have any nice name like everything else. It's a very generic name. This is going to use magnesium or some other divalent metal to help with the charges. Interestingly enough, this enzyme is inhibited by fluorine. So if you inhibit this enzyme, you more or less stop glycolysis inside the cell. So if you want to kill something, fluorine will do it. And that's how fluorine can kill you. Fluorine poisoning, eat enough fluorine and you're going to stop glycolysis in your body. Luckily, the amount of fluorine they put in the water is way, 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 way lower for harm of us. We don't need to worry about that. So PEP, we've seen PEP before on our table of phosphate compounds. PEP is another compound that has more energy than ATP. So you can probably guess where we're going here. We're going to use this to make ATP. Because you remember with our phosphoryl transfer energies that we talked about, I think last week. If we have a high energy phosphate compound, we can transfer our phosphate to lower energy compounds spontaneously. So this PEP has a lot of potential energy built into it that we're going to use to make ATP. And that's being shown here. Pyruvate kinase, lovely name. Pyruvate is the molecule. Kinase, I'm working with phosphates. So PEP, very high energy. Again, high energy. We're going to use that high energy compound to make ATP. So we cleave that phosphate, put it onto ADP, and make our ATP. And this mechanism is very similar to mechanism of our first reaction, where we have magnesium. Magnesium is going to go and help stabilize all the charges, right? Magnesium is +2. It's going to go interact with all these negative charges and allow for the transfer of the phosphate from the PEP, which is shown right here, to an intermediate enol pyruvate, and then finally pyruvate. So very simple mechanism. Take a phosphate from PEP, you make ATP. You have an intermediate state, enol pyruvate. We need to get rid of that CC double bond. So we add an acid and we make pyruvate. And that is the end of glycolysis. Really simple, right? 10 easy reactions I'm sure you have down, no problem already, like back of your hand. I know it's like, it is really complicated, especially the first time you look at like glycolysis, and it's probably very overwhelming, but try and work through it step by step, thinking about the logic, always trying to remember where we're going to, what's our end goal, what are we trying to do at each step. Hopefully that'll make it easier to learn once you understand the chemical logic behind it. So I already answered this. Why does this reaction have a large negative delta G? PEP is very high energy, higher energy than ATP. So moving an phosphate to ADP releases energy, because PEP has a an energy of like -60. That's how much energy you get out from removing the phosphate. ATP is like -37. So it's free. We're using that energy of cutting the phosphate from PEP to put it on ADP. And that's a free process for us. As long as we have an enzyme to help with that. It's going to be spontaneous with the help of pyruvate kinase. And you can see it's not really reversible. There's no bottom arrow on this. This is large, this is negative, this is irreversible. Remember, we talked about these large delta Gs, these are going to be control points. We cannot go back. Once we make pyruvate, we're stuck in pyruvate. We cannot go back to make PEP. We can't go back up using the enzymes of glycolysis. And we're done with glycolysis, the second stage. We took we created 2 pyruvates, 4 ATPs, and I should also say 2 NADHs. Because although they're not energy yet, as long as you breathe oxygen, they will be energy. So glucose goes in. What we create, 2 pyruvate, 2 NADHs, 2 ATPs, 2 water molecules, and 4 acid molecules. NADH, like I said, electron transport chain. ATP, we can use in any way we want. Pyruvate, we have many different fates of pyruvate that we'll begin to look at today. Oxidative phosphorylation being the most energy rich of that process. But with that, is there any questions anybody might have about glycolysis itself? Yeah, mnemonics definitely help with all the enzymes. Some of my students last semester or 2 semesters ago, they found some creative ones that I cannot repeat on video. For some reason, they wanted to tell me all about them. Yeah, I'm not going to have myself on video saying those words, but yes, glycolysis is used in like, the MCAT, everything. So look up mnemonics for the enzyme names, for the molecules names, and I'm sure you'll get some interesting ones. Myself, I wasn't smart enough to do that, and I just brute forced it when I was a student. Lots of flashcards. All right. So which reactions are irreversible? So we already kind of mentioned that, but the only payoff reaction, and the second step is a payoff step, is the last one. And it's kind of shown here in our image. You can see everything has a double arrow except the very last reaction. So only reaction 10 of stage 2 are irreversible. If you want to go back to the previous steps, 1 and 3 are irreversible, and 10. So 1, 3, and 10 cannot go backwards. Their delta Gs are too high. That ends this PowerPoint. So let me switch PowerPoints here so we can talk about the fate of pyruvate now. And just as a way to refresh yourself, since we covered glycolysis over like 2 different sessions, take a minute or so, and you don't have to answer in chat. Just take a minute or so and remind yourself of this. What did we make in the preparatory phase of glycolysis? What do we make in the payoff phase? So a good little review here for you. So I'll give you a minute or so to review your notes and remember all that. So in the prep phase, we created 2 phosphorylated 3-carbon compounds, GAP, at the cost of 1 glucose. Well, I mean, 1 glucose is what we invested at the very top, plus 2 ATP. Payoff, we get 2 pyruvate out and 4 ATPs and 2 NADH. So in a nutshell, that is glycolysis, the important bits. So now let's see what happens to pyruvate. There's 3 different things that can happen to pyruvate. If you have oxygen available, the aerobic pathway, you can go through the citric acid cycle and your pyruvate is broken down to carbon dioxide and water. If you do not have oxygen available, like if you are using your muscles, then you can do what's called homolactic fermentation, where you take your pyruvate and make it into lactate. And the reason to do this is you're going to just use the NADH that you created to remake NAD+. Yeast can do something else. They can take their pyruvate and make ethanol and carbon dioxide. Now other microorganisms can do that as well. That's how they will recover their NADH. They will turn that back into NAD+, make ethanol. Animals, we can't do that, which I guess is a kind of a good thing, because imagine that you're working out like really hard and then you just like feel drunk and now, oh no, I can't do stuff anymore. Or if you want to talk about evolution, let's say you're running away from a tiger, you're using your legs a lot. Oh no, I used my legs too much. Now I'm drunk because I made ethanol. So it's probably a good thing animals can't do that. Maybe a bad thing now in 2021 when we don't have to run away from tigers, but it was not a great thing back then. So thinking about this in a big picture, why would we take the NADH we just created in glycolysis and immediately make NAD+ with like no benefit? Because remember, NADH is an electron carrier. We can use NADH to do a lot of things, mainly to make ATP. Here, we're just immediately throwing it away and saying, those electrons you had, give it right back to pyruvate and recreate NAD+ for me, please. What is the purpose of that? Why would we ever do that? And think back to glycolysis. And think back to when we would do this. So that's how we can really answer this. When would we do this? Like under what conditions? And yeah, under what conditions would we do this reaction? And then think about, why is that important overall on a big picture? Anyone want to take a stab at that? All right, so let's take a look at this logic then. So when do I want to create pyruvate to lactate? I would guess because you don't have to keep putting NAD+ into glycolysis. You got some right keywords in there. Let's try and put those together into a big picture though, right? So pyruvate goes to lactate when we are low in oxygen. We don't have oxygen around. As animals, when don't we have oxygen on the cellular level? That happens when we are maximum effort of muscles. We are doing something that uses our muscles a lot, okay? So if we're at maximum effort of our muscles, generally speaking, you want to keep up that effort as much as you can. You're using your muscles for some reason, so you would not want your muscles to just stop working. So you want to keep this up. You want to keep up maximum effort. And hopefully you remember from Biochem 1 how muscles move. with myosin and actin, the power stroke. Remember, what does the power stroke use? Power stroke uses ATP. So you're using ATP to move. So you need to remake ATP. But you don't have oxygen. And one of the ways you make ATP is through oxidative phosphorylation. So you can't do oxidative phosphorylation. That's impossible right now. So the only way to get ATP is through glycolysis. That's the only thing available to you under these conditions. If you remember some of the steps of glycolysis, they take NAD plus to make NADH, right? However, at the very end of glycolysis, you aren't making NADH, but we can't use that because we're not doing oxidative phosphorylation. And if we do not replenish the amount of NAD plus in our cells, glycolysis can't move forward. You need NAD plus or glycolysis doesn't work. Glycolysis stops working, your muscles stop working. Your muscles stop working, depending what situation you're in, you're probably dead. You don't want that to happen as an organism. So going from pyruvate to lactate is a smart way to recycle this NADH back into NAD plus. So you can make NAD plus through NADH through glycolysis. Through glycolysis, you make ATP. Through ATP, you keep your muscles working, and hopefully you stay alive. So does that logic make sense to people? Why we're going through the effort of making lactate it's more or less just to get rid of NADH because we don't get any energy out of it. Or any questions about that logic, I should say. All right, so we'll move on. Hopefully, with all this knowledge, you're starting to appreciate how amazing one single cell is, that all of this is going inside your bodies right now. This whole web, and we are barely scratching the surface of complexity of the cell. All right, so let's take a look at this a little more in depth. Basically, this slide has a lot of what I just said. Muscles are working, demand for ATP is high, therefore oxygen is low. We can make ATP via glycolysis. This is way faster than oxidative phosphorylation. So when we're working, this is a way to go. The enzyme that does this is lactate dehydrogenase. So lactate, first off, dehydrogenase. This means like it's going to be redox. NAD is probably going to be available. It's named for the reverse reaction. It's named for lactate going to pyruvate because this is a reversible reaction. So it's named for the reverse reaction of what we're studying. So LDH or lactate dehydrogenase is the enzyme that's doing this. And basically what we're doing is we're taking our glucose and making 2 lactate plus 2 ATP. That is how we can make energy quickly when we need it really fast. Lactate, we can't do anything with lactate. That's why I say it's a dead end. Metabolically speaking, it's over. Once you're at lactate, you can't do anything with that 3-carbon compound. So what you need to do is you need to kick lactate out of the cell. And what happens is that lactate will eventually go back to your liver. And inside the liver, you will spend energy to remake that into glucose. Then your liver will either store it or it will send it back as glucose to be used as energy. And we'll look at this cycle later in the semester, but I just want to bring it up here since we're talking about it. It's a cool little cycle inside your body where you go from muscle, right? So you're using your muscles and you take glucose and you make it to lactate, right? And then you spit that out of your muscles and it goes to your liver. Your liver takes that lactate, makes it back to glucose, sends it back to your muscle. That way you can keep reusing the lactate to make more energy. Granted, you are using energy in the liver to do that, but when it comes like, hey, I'm using my legs a lot to run away, that's fine. You can, liver, use your energy. I need to keep my legs moving. So that's a way we can keep our muscles going in a prolonged state. So if you're jogging for like an hour and a half, this is one way we can keep the energy up. Your muscles are burning, you have that lactate, send that lactate to your liver, remake glucose, send it back to your legs. Interesting. This is one of those misconceptions, the general populace. When you talk about muscles burning, they say, oh, all that lactate. Lactate doesn't do anything. It doesn't make you sore. The reason why it makes you sore to do this is because if you look at our reaction here, we are having acidic conditions. So what's happening, and it's in glycolysis as well. When you're going through glycolysis a lot, when you're doing this, you have acids building up naturally. And since you're doing glycolysis really fast, you have more and more and more acid building up. So lactate doesn't hurt. Glycolysis hurts when you do it very fast. So it's due to the buildup of acid is why we have that muscle soreness and fatigue. So any questions about homolactic fermentation? I guess I can re-put that there in case you're still writing. All right. So yeast, they do something a little bit different. Let me redraw or rewrite the things here. Pyruvate. This is acetyl aldehyde. This is ethanol. And this is, we're just going to call it TPP because I'm not going to rewrite all that. So yeast do something a little different. They'll take the carboxylic group from pyruvate and they'll kick it off to make carbon dioxide and make ethanol from that. So if you ever seen like production of alcohol, it's always a very gassy process because to take this pyruvate to ethanol, you let off carbon dioxide. It's a two-step process. You take, the first thing you do is pyruvate decarboxylase. Again, lovely name for an enzyme, tells you exactly what it's going to do. It's going to work on pyruvate and is going to remove the carboxyl group, so pyruvate carboxylase, to make acetaldehyde and carbon dioxide. The reason we can't do that is we don't have this enzyme. We do have the next enzyme, though, is that you take this acetaldehyde by alcohol dehydrogenase. Here, we are re-getting our NAD+, and we are making ethanol. For this to work, yeast need thiamine pyrophosphate, or TPP. This is a cofactor, and if you remember what cofactor means from Biochemistry 1, it's a molecule that is naturally not part of the enzyme, but needs to be incorporated into the enzyme for the enzyme to work, right? So it's not, so TPP is not like in the DNA of the organism to make that with the enzyme. We have to take another molecule, in this case, vitamin B1, and what yeast will do is they'll take this thiamine or vitamin B1, make it into TPP, and now that TPP can be associated with pyruvate decarboxylase and do this chemistry. And that binding is all non-covalent. So when TPP binds, no covalent bonds are formed, all non-covalent interactions. So hydrogen bonds, ionic bonds, hydrophobic bonds, all that stuff. So that's how pyruvate is used in yeast and other microorganisms. And it's like the same idea. Remake that NAD plus so we can remake glycolysis. So another good critical thinking question for people. Pyruvate to lactase, readily reversible. We can go one to the other. Pyruvate to ethanol, not reversible. Why? Why is one reversible? Why is the other one irreversible? Would it be due to the effects of keeping ethanol in the system? I don't know, but probably not. If it is, it's very minor. There's something else that's happening. I guess another way to think about this, to go back from ethanol to pyruvate, what steps were mainly different from going from lactate to pyruvate? You see they both have NADH and NAD+, but what is the big difference between how animal cells use pyruvate and yeast cells use pyruvate? What was the big step that's different? Pyruvate decarboxylase. Yes. What's happening in pyruvate decarboxylase? What are we getting rid of? That carbon dioxide, right? So the reason why that's the second one's not reversible is that we're breaking a bond to make a gas. We're breaking this bond off and we're creating a gas. This CO2, once it's in a gas, it's just going to fly off because that's what gases do. CO2 is not dissolvable in water. So what could happen is that it could turn into carboxylic acid or whatever. It's going to go away. So if I wanted to go reverse, I would somehow have to capture a CO2 molecule and put it back in and jam a CO2 molecule onto pyruvate. That would probably cause energy. And it's also the complication of how do I get this gas captured, right? Because that gas is gone. So it's going to be really hard to do to recapture that CO2 and work in reverse, because you probably need to put in some energy as well. Well, for our first reaction, the only thing that's really moving around is, well, a hydrogen, right? And since these reactions are happening in water, hydrogens are everywhere. You can easily get a hydrogen. So we can just grab a hydrogen, put it back on to NADH. We're good to go. Here, for pyruvate decarboxylase, want you to remove a CO2. And we're going to see this in glycolysis. Animal cells use something like pyruvate decarboxylase in different, and they do it right before the citric acid cycle, where we do like the same exact reaction. Once that CO2 is gone, it's gone. Does that make sense to people, or any questions about the chemical logic about that? All right. moving on then. So let's take a look at the energies of this. Standard energies again. To make lactate minus, just say 200, minus 196, minus 200. Make ethanol minus 235. So you do get a little more energy to make alcohol than you do for lactate. But no matter what process you go, really, you're just making 2 ATP because this process you're redoing to go through glycolysis, and you're going to be net 2 ATP. Overall, making 2 ATP costs you 61 kilojoules. So if you want to think about this in money terms, right, for human, our human cells, like if you have $200, you're going to go and buy something worth $61 and you don't get change. You get the cashier 200, they give you the ATP worth 61 and they keep the other 139 kilojoules or $139 for themselves as a tip. So this is really inefficient to do. You're looking at less than 50% energy efficiency, a little bit greater than 50% around that mark. All that other energy is heat. So it's gone. Once you create that ATP, you can't do anything. You have to remake that lactate into something else. So not the first choice for making energy because you're wasting so much of the potential glucose doing this, but it does have its uses under some circumstances. Way less efficient in oxidative metabolism. You're making 32 ATPs, but when going to make lactate, you are going 100 times faster. So in the time it makes 32 ATPs using oxidative methods, you're making 200 ATPs through going to lactate. You're wasting a lot, but you're getting a lot faster. So when you're using your muscles, you're more or less doing this. You're doing this anaerobic respiration, remaking that ATP incredibly fast at the cost of wasting a lot of potential energy of that glucose. So the last question that I have for you this week is we're going back to some electrical chemistry because I know how everybody loves the Nernst equation. So when we look at lactate dehydrogenase, I give you the standard potentials of pyruvate going to lactate and NAD plus going to NADH. I want you to tell me what the delta G of this reaction is at a pH of 7 under 2 sets of conditions. If the ratio of lactate to pyruvate is 1:1, and the ratio of NAD plus and NADH is 1:1, versus if the ratio to lactate to pyruvate is 160:1, and the ratio of NAD plus and NADH is 160:1. So I'm just going to rewrite A and B because it always confuses people. When I say that this ratio is 1, that means lactate and pyruvate are the same number. NAD plus and NADH are the same number. When I say this ratio is 160, that means lactate's 160, pyruvate's 1, right? 160 divided by 1 is 160. Same thing here. So see if you can at least start to get this going in the next couple of minutes. And what I would do is like after 3 or so minutes of you struggling, I will come back and do A, and then I'll let you struggle on your leisure with B. But yeah, take a couple minutes, see if you can do this. If you have any questions, do let me know. And just as a reminder of what the actual reaction is, remember our reaction is pyruvate plus NADH is making our lactate plus NAD plus. Right, because that's the whole purpose of this, is to remake that NAD plus. And one of the things we need to solve this is our delta E. And remember, delta E is what's being reduced minus what's being oxidized. So our pyruvate is being reduced, our NADH is being oxidized. And even though, and this is something we talked about when we first looked at Nernst's equation, you always use the reduction potentials. You don't flip those like at any point, even though in the equation, like this NADH is actually our reactant. In the half potentials we have written here, you don't need to change those numbers. So in our equation here, the pyruvate is being reduced. So that is negative 0.185 minus a minus 0.315. How do you know which one is reduced and which one's oxidized? That has to do from looking at our real equation, right? So our real equation is inside the body, right? What are we doing? We're taking pyruvate to lactate, and we're converting NADH to NAD+. And what I do is I look at our reduction potentials. Remember, every time we write a half reaction, we are writing it as if the molecule is being reduced. So here, if pyruvate gets reduced, it makes lactate. So if pyruvate is being reduced, it should be a reactant. It is a reactant. If NAD plus is being reduced, it should be a reactant. But in our actual equation, it's not. It's a product. Therefore, when we're looking at the real chemistry, reaction 2 with NAD plus is actually opposite than what's going on in reality. So that means it must be being oxidized. Another way to do it is just look at the reactions and remember how NADH and NAD plus works. And it's even written here, right? NAD plus 2 electrons makes NADH. So if our reaction was instead NAD plus making NADH, and I'm just going to say A going to B, 2 random molecules. If it's written like this, NAD is being reduced to make NADH, A is being oxidized to make B. That's not what's happening. Here, NADH is being oxidized to make NAD plus, and if one pair of molecules are being oxidized, the other pair of molecules have to be reduced. So those are a couple of different ways which you can go about understanding or looking at what's being oxidized or what's being reduced. That make a little sense of how I figured that out? All right. So we calculate delta E, and this is actually delta E. Whoops, don't want my eraser. Delta E not prime. And that will give us a positive number, which is 0.130 volts. All right. Now we can set up our delta E equation. Delta E equals delta E not prime minus RT and F ln products over reactants. So this is our Nernst equation, ln of products over reactants. Our products are lactate, NAD plus. Oh, I did not give you an R or T. So it's very hard to do this reaction if I don't give you one of those. We're going to say T is 25C. And we are going to say R for joules per mole K. So I solve for delta E because I have an equation. I have all my variables. So we already solved for delta E not prime, 0.130 minus R. And we want this in 8.314. This actually might be the wrong R for this equation. Nope, it's the right one. It's the right one. Times 298 divided by N. Remember, N is the number of electrons. Here, we're using 2 electrons. So it's 2 times Faraday's constant, 96485 multiplied by natural log. If everything is 1, whoops, I forgot the denominator. So products over reactants, pyruvate, and ADH. Everything is 1, it's the natural log of 1. How do you know when to use A, E, or AB? EA and EB mean the same thing. It's just saying molecule A or molecule B. It's the same reaction. This is just saying, like, in our reaction, we have 2 half reactions we care about, which is what we have here. We took our big reaction, and we can split it into half reactions. We can calculate the electron potential of each half reaction. And we use this information to figure out overall, when I combine these two, how much electricity is being generated. So EA, EB, same exact thing. What's important to remember is that you need to take what's being reduced in your equation minus what's being oxidized to get overall delta E. Right. And I know I'm all out of time here, so let me just finish this super quick. When you do the natural log of 1, the natural log of 1 is 0, right? So delta E equals 0.130 because natural log of 1 is 0. So that whole multiplication term cancels out. We can then calculate delta G by using our equation on the bottom left there. Delta G equals minus NF delta E. So N is 2, F is the constant. Delta E we just calculated is 0.130. And that should be minus 25.1 kilojoules per mole. So I know using the Nernst equation is super confusing because there's a lot of moving parts and a lot of unknowns you have to declare. See if you can figure out B. Give it a stab with B. Before I let you go, is there any more clarification that you would want on A? How I decided stuff or anything like that? How do I know lac and NAD go on top? Again, that's why I rewrote the actual equation that's happening, because when you do natural log, it's always products over reactants. So lactate and NAD plus are my products. Pyruvate and NADH are my reactants. So whenever doing any equation in chemistry, but especially the Nernst equation, write out the real reaction that's happening. That'll help guide you. It'll help guide you on what's being reduced and oxidized. And when you go to calculate delta E, it'll tell you what goes on top and what goes on bottom of the natural log. Any study guides that will be available? I have not made study guides for Biochem 2 because all I did for Biochem 1 is just go through my slides and just ask random questions. I have not done that for Biochem 2, so not right now. It would be good practice for you to make your own study guides, I think. It'll be a great skill to be able to hone. Looking at my slides, my videos, and trying to pick out what are the most important points of that slide. That's what I used to do as a student anyways. Those study guides that I posted for Biochem 1, those are what I did for myself when I was a student. I made those all the time. So I was just trying to show you like how I studied. So yeah, short answer, no, they are not available. Trying to make you a little more independent. All right, with that, as always, I will put a homework up. And that's all for this week. Hope you learned a bunch. If you have questions at any time, please let me know. Otherwise, I will see people on Tuesday. Have a good weekend, everybody.